Greetings, everyone. I'm sorry about the low volume here, and if there are any noises that come in from outside, I'm recording in a public space today. Today I want to delve a little bit deeper into the nephron and exactly how it works and how it functions. So, if you recall, we started the nephron off at the glomerulus. And if you'll recall, blood from the renal artery and the renal arterioles is going to flow into the glomerulus through the afferent arteriole and out of the glomerulus through the efferent arteriole. Inside the glomerulus, you have a bunch of capillaries. These capillaries are fenestrated, meaning that there is, it's very easy for fluid to exit the capillary. There are 4 forces that affect filtration in the glomerulus, 2 that increase it and 2 that decrease it. And these are called the Starling forces. Okay. And the first Starling force is the one that we have talked about many, many times, and that is hydrostatic blood pressure. So let's go hydrostatic blood pressure. There we are. So what hydrostatic blood pressure means is that's just normal blood pressure. It's the actual force of the fluid in blood on the walls of the capillary. And because our afferent arteriole is larger and our efferent arteriole is typically smaller, you have a very, very high blood pressure inside of the glomerulus, and that will tend to drive fluid out. And that fluid we're going to call filtrate. So hydrostatic blood pressure is going to drive fluid out. The higher the hydrostatic blood pressure, the more fluid you're going to drive out into Bowman's capsule. So what are some things that might increase hydrostatic blood pressure? Well, anything that increases like your body's net blood pressure, all of the things that we talked about in the blood pressure section, so things like blood volume, viscosity, blood vessel diameter, things like that. Those all continue to be relevant. But we have, in the glomerular space, we have some particular things, which is the size of the afferent relative to the efferent arteriole. If you dilate the afferent arteriole, you have more blood going in, same amount coming out, blood pressure rises in here. Same if you constrict the efferent arteriole. If you constrict the efferent arteriole, you have the same amount of blood going in, less going out, blood pressure rises in here. The higher blood pressure rises in here, the more fluid you squeeze out of the capillaries. Okay, so what is the second force? The second force is colloidal. Colloidal, basically osmotic pressure of the blood. So colloidal osmotic pressure of the blood. This blood is losing a lot of fluid, and it's not losing protein, and it's not losing blood cells. If you recall, protein and blood cells are the things that are going to make your blood viscous. They're particulate matter. They are solutes, and so that means that you have a lot of fluid, a lot of water in the filtrate, and less water in the capillaries. So osmotic pressure is going to pull water back in to the capillaries. So these, I'm going to just put like a little red dot after here to let you know that hydrostatic blood pressure is the red. The colloidal osmotic pressure, I'm going to do with blue, and that's actually going to pull fluid back in to the capillaries. Now what I want you to keep in mind is that hydrostatic forces push water out, osmotic forces pull water in. These are the two normal forces. They actually operate in every capillary, right? If you have like a normal capillary bed where you have an arteriole, all right, and it branches off into a capillary system, eventually converging into a venule. We talked about this in the body. Remember that so in your arteries, you have high blood pressure, and so in your arteries you will have the high blood pressure forcing fluid out into the external area, into the interstitial fluids, and that is lymph, right? The fluid that's forced out of the blood vessel is lymph. Now over on the venular side, you have much lower pressure, but since you've excluded water, you have much higher osmotic pressure, and that's actually going to pull fluid back in to the capillaries and venule, and about 85% is reabsorbed. And the other 15% becomes the lymph that collects in your lymphatic vessels. So this is actually a concept that we talked about before when we talked about the lymphatic system. But here we're talking about it in a special context of the nephron, and the fact that it's taking place inside of the glomerulus, where we have fenestrated capillaries, means a few things. First off, the quantities of fluid that we're talking about are going to be simply much, much greater, because it's much easier to force water out because blood pressure is so much higher. Secondly, this is an enclosed system. This is a capsule, and it's possible to fill that capsule so much that it actually exerts back pressure. So that's going to be our next force, is hydrostatic back pressure. of the filtrate. And we'll do this in green. And the hydrostatic back pressure of the filtrate, that's going to be once you've filled up Bowman's space, you try to squeeze more water out. Well, that water can only drain so fast, so you're going to increase the pressure and actually squeeze some of the filtrate back in. So that's going to be a force that forces filtrate back in to the capillaries. Our last force is going to be colloidal osmotic pressure of the filtrate. The filtrate is not water. It does have stuff dissolved in it, salts, sugars, a whole bunch of things like that. And so it's going to, let's do this in yellow here. It's going to act to pull water out and into the filtrate. Now, what really matters here is actually the difference in the colloidal pressures and the difference in the hydrostatic pressures. Because under a normally functioning kidney, the colloidal pressure of the filtrate should always be lower than the colloidal pressure of the blood. After all, it's made from the blood, and you're squeezing out most of the water, so it's always going to have more water in it than the blood does. Similarly, again, under a normally functioning kidney, your hydrostatic blood pressure should always be higher than the hydrostatic back pressure. You know, how could your back pressure get higher than the pressure that is creating it? There is a circumstance which we may talk about in a later lecture. But generally speaking, in a properly functioning kidney, your blood pressure should always be higher than the back pressure. These are the four Starling forces that go into making your glomerular filtration rate, which is how much filtrate you make from the glomerulus. So these are our Starling forces. Just to sort of go through them in a little bit more detail. Blood hydrostatic pressure, that's your blood pressure. But specifically, it's the blood pressure inside the glomerular capillaries. And that blood pressure is usually a lot higher than in the other capillaries of your body. So in the glomerular capillaries, we're talking like 60 millimeters of mercury pressure, compared to about 10 to 15 in most of the other capillaries of your body. So you're filtering fluid out, filtrate out at a much higher rate because of the higher pressure and because of the fenestrations in the capillaries. The hydrostatic pressure in the capsular space, this is going to depend upon how much filtrate you're making. The faster you make filtrate, the faster the capsular space builds up, the more back pressure it exerts. It's also going to depend on your rate of urine drainage. If, for some reason, your kidney gets blocked up, for instance, with a kidney stone, then that filtrate won't be able to drain away, and that back pressure can build up dramatically. Now, again, that's a pathological state. But the hydrostatic pressure in a normally functioning kidney is usually somewhere around 18 millimeters of mercury. So significantly significant. It's a real number, but it's always should be lower than the BHP. The colloidal osmotic pressure of the blood should be the same in the glomerulus as everywhere else, about 32 millimeters of mercury. That's pretty normal, but it can change. If you're dehydrated, then your colloidal osmotic pressure is going to go up. If you have a lot of blood proteins or a lot of blood cells, maybe you've been EPO doping or something and you've just got a bunch of red blood cells or something like that, then the colloidal pressure of your blood could go up. It's usually about 32 millimeters of mercury. The colloidal pressure of the glomerular filtrate is typically negligible. It doesn't have very much protein, it doesn't have any cells, so its colloidal osmotic pressure is typically 0 in a properly functioning kidney. Now, in a kidney that's undergoing glomerular nephritis, or that has damage to it that's allowing blood and protein to leave the glomerulus, that can change. But under normal circumstances, we're going to consider this glomerular filtrate osmotic pressure to be 0. And what you're looking for is the net pressure. Remember, hydrostatic pressures push, osmotic pressures pull. So that 60 is forcing blood out. That 18 is pulling blood back in. And that 32 the 18 is pushing blood back in. That's your hydrostatic pressure of the capsular space. And that 32 is pulling blood back in due to osmotic pressure. So the 60 minus the 18 minus the 32, that's your net out pressure. So in this case, in a normally functioning kidney, it should be somewhere around 10. That can change dramatically based on how much blood pressure you're generating in the glomerulus, what the osmotic pressure of your blood is, and the hydrostatic back pressure of the capsular space. This one usually doesn't change under normal circumstances. Those Starling forces will determine the amount of pressure that's forcing blood out of your glomerulus. As long as it's positive, you will have a glomerular filtration rate, which is the amount of filtrate that you're forming per minute in both your kidneys. A typical glomerular filtration rate is your net filtration pressure, that's what we just calculated on the last slide, times the filtration coefficient, which is dependent upon the amount of surface area you have, the permeability of your glomerulus. This can be dependent upon drugs and hormones. It can be dependent upon how much damage your kidney has taken over your life. But in males, this should typically be around 180 liters per day. In females, around 150 liters per day. That's how much filtrate is made by your kidneys. You don't have 180 liters of blood. You don't have anywhere close to 180 liters per blood. So all of your blood is filtered multiple times per day. Obviously, most of that stuff is going to be reabsorbed, which we're going to talk about next. So you usually produce about 50 to 60 times the amount of blood that you have in filtrate. 99% of that's going to be reabsorbed. Under normal circumstances, you're only producing about 1 to 2 liters of urine per day. I'll remind you, this is the glomerulus. We talked about this before. The glomerulus is divided into 5 portions, or sorry, the nephron. But this is the nephron. This is divided into 5 portions. The glomerulus, that's where filtration happens. Proximal convoluted tubule, the loop of Henle, distal convoluted tubule, and collecting duct. A single collecting duct will be shared by multiple nephrons. There are actually 2 different types of nephrons that you'll find in the kidney. The more common one are what are called cortical nephrons, which you see up here. There's about 85% of the nephrons in your kidney, and they're called cortical nephrons because the glomerulus is actually located pretty high in the cortex. So you see here, these are your arcuate artery and vein. They mark the boundary between your cortex and your medulla. And so you can see that the glomerulus here is located pretty high up in the cortex. That means that when the loop of Henle dips down, it's only going just a little bit into the medulla. It's only got this small portion at the bottom here that's going into the medulla. Most of the loop of Henle stays up in the cortex. The nephron loop is actually pretty short, or the loop of Henle is actually pretty short. And your efferent arteriole, which you can see exiting here, branches into a whole bunch of peritubular capillaries that surround the proximal and distal convoluted tubules. Cortical nephrons typically produce dilute urine. Here we have a juxtamedullary nephron. Juxta means nearby or next to. So what this literally means is a nephron that is next to the medulla. And so you can see here, this glomerulus is located right above the medulla. So this, again, has a proximal convoluted tubule coming out, and it has a loop of Henle that dips way down into the medulla before coming back up to the distal convoluted tubule. The efferent arteriole branches into what are called the vasa recta. They're similar to peritubular capillaries, but they have different permeabilities, and we'll talk about that later. These are going to maintain the salinity gradient of the medulla. These are ones that are going to take a lot of water out on the way down and take a lot of salt out on the way up. They tend to produce more concentrated urine and conserve more water. So we're going to go through the steps of urine formation, right? The first is filtration. Filtration happens in the glomerulus. The amount of filtrate that you produce is determined by the Starling forces, like we just talked about. The second step of urine formation is reabsorption. Reabsorption happens in pretty much the entire rest of the nephron, but different things are absorbed in different areas. Most of the reabsorption of non-water stuff happens in the proximal convoluted tubule. Water is absorbed throughout the entire process, and other ions are absorbed in salts are reabsorbed in the ascending loop of Henle. Calcium, magnesium, some other salts are absorbed in the distal convoluted tubules. So you have reabsorption happening pretty much all down the rest of the nephron. Tubular secretion. This also happens pretty much everywhere in the throughout the nephron, although there isn't a whole lot that happens in the loop of Henle, but this is where you're going to use active transport to pump stuff in to the filtrate. Extra stuff that you want to get rid of. So here you can see you're reabsorbing stuff from the filtrate into the capillaries, and in secretion, you are pumping bad stuff, toxins, junk like that, from the capillaries into the filtrate. Last, you have water conservation, pulling water out of the filtrate to concentrate it down. And you always remove a lot of water. Sometimes you remove more than others. And the big difference in, like, you're always going to be removing a lot of water in the proximal convoluted tubules. You're always going to be removing a lot of water in the loop of Henle. Whether or not the collecting duct is removing water is one of the main things that controls whether you're producing dilute or concentrated urine. In the proximal convoluted tubules, most of the glomerular filtrate is reabsorbed. All right, this is where you've got a whole bunch of glomerular filtrate, and reabsorption is fairly easy. What you're going to, there are lots and lots and lots of microvilli on these cells. So the cells of the proximal convoluted tubule are mostly like cuboidal epithelium, and they've got a brush border of microvilli to increase surface area. And all of that increased surface area is necessary to get a very fast reabsorption rate. You're also going to just spend ATP like there is no tomorrow. So, like, your proximal convoluted tubules alone account for about 6% of your resting ATP and calorie usage. And you're going to be reabsorbing, you don't directly reabsorb water, but water tends to follow salt. So you're going to be reabsorbing salt. You're going to be reabsorbing nutrients, sugar, amino acids, vitamins, everything like that. And it's going to be reabsorbed through two routes, the transcellular route. In the transcellular route, what you have is your solutes, which I'm going to do here in green, are going to get absorbed into the cells and then through the cells into the blood beyond. That's the transcellular route. There's also the paracellular route. These cells, these epithelial cells of the proximal convoluted tubule, they don't have tight junctions. They have fairly leaky junctions, and so some things can just passively, and I'll do this in yellow, can just passively diffuse between the cells and back into the blood. A lot of water goes by that way. And as the water flows back in through this paracellular route, it causes what's called solvent drag, which is just that the water carries whatever is dissolved in it with it as it goes in. So you're going to be like pumping salt and sugar back in through the transcellular route, and then water is going to follow, because where salt goes, so goes water, and that water is just going to carry whatever happens to be in it back into it. Potassium, magnesium, phosphate are carried through the paracellular route with water, sort of passively. phosphate is also co-transported with sodium. So you're absorbing sodium, and that's going to pass through the transcellular route with potassium, or with phosphate, I should say. Some calcium comes back in through the paracellular route. That's the amount of calcium that you reabsorb no matter what. But most calcium reabsorption actually occurs later on in the distal convoluted tubule and will be controlled by parathyroid hormone. Glucose is also co-transported with sodium by the sodium-glucose transport mechanism, and the sodium that's out here that's being reabsorbed is actually not just the sodium that was in the filtrate, because you have a sodium-potassium ATPase pump that's constantly pumping sodium out and potassium in. Potassium channels are leaky. If you recall, remember when we talked about muscles and most particularly nerves? Potassium channels are leaky, so the potassium can leak back out to be re-imported in. Sodium channels are not leaky, so you're pumping all this sodium out. The only way the sodium can come back in is if it carries a glucose with it. That's symporting. So you're not just depending upon the natural amount of sodium in your filtrate. You're actually spending ATP to create a sodium gradient and pull the glucose back in. Normally, all of the glucose, all of the amino acids, all of the ketones are going to be reabsorbed unless the system gets overwhelmed, which is the case in diabetes. And this basically is just what I was talking about there. Sodium reabsorption creates an osmotic and electrical gradient that drives the reabsorption of water through the paracellular route, which carries other things with it: magnesium, phosphate, potassium, that sort of things. Now, sometimes, depending upon what your blood pH is, if your blood pH is too low, meaning too acidic, you need to get rid of hydrogen ions, acid. In that case, you will have a sodium hydrogen antiporter. It's going to pump sodium back into the cell and hydrogen out. If this antiporter, there are hormones that make this antiporter more active, and the more active this antiporter is, the more acidic your urine becomes, and the more basic your blood becomes. So tubular secretion. We already talked about tubular secretion of one thing, acid. We saw that that was done by an antiporter. By letting something in, you can force something else out. That's going to be a normal way that we have tubular secretion. The tubular secretion happens all throughout the nephron, but it is particularly common in the proximal and distal convoluted tubules. Secretion in the proximal convoluted tubule and nephron loop maintain acid-base balance by pumping hydrogen out into the urine, making the urine more acidic. Under certain circumstances, if your blood is too basic, you can actually pump bicarbonate out, making your urine more basic and your blood more acidic. That's pretty rare, simply because your body generates a lot more acids than it does bases. So dealing with acidic blood is simply much more common than dealing with basic blood. Waste products. Now, hopefully, a lot of these things were just eliminated in the filtrate, but we're not going to just depend on the filtrate. We're actually going to actively transport a lot of these waste products out into the urine as well. Urea, uric acid, both of those are from the breakdown of proteins. Bile acids, which we talked about from in the digestive system. These are breakdown products of cholesterol. Ammonia, again, that's a breakdown product of proteins and some other toxins that are destroyed in the liver, and a little creatinine. Generally speaking, if you have a lot of creatinine in your urine, that can be indicative of liver failure and kidney failure. So, also drugs and contaminants. Morphine, penicillin, aspirin, lots of other drugs are actively transported, secreted into the urine. So the nephron loop, or the loop of Henle. Remember, this is going to go basically down into the medulla and then come back out of the medulla. And generally speaking, on the way down, we have what's called the thin segment, which is made mostly of squamous epithelium. And then on the way up, you have the thick segment, which is made of more cuboidal epithelium. And the reason for this is that on the way down, water is passively diffusing out. It's through osmosis. And so you want to keep the amount of distance that it has to transfer as thin as possible. So you want to have squamous cells. You want to have as few barriers between the water and getting out as possible. So you keep it very thin so that the water can easily diffuse out. Let's draw some water diffusing out. We'll put the water in blue. On the ascending side, you're going to be actively transporting salts out. We'll do the salts in green. And since you're actively transporting the salts out, it doesn't matter how thick the cells are, because they're being kicked out using energy. And this stuff that's being kicked out here is primarily going to be sodium, potassium, and chloride. They're actually leaving by both. So while water leaves only by passive diffusion, these salts are leaving by both active transport and diffusion. There is actually a gradient, right? So they will leave on their own, but they don't leave fast enough on their own. So you're going to spend some energy to kick them out even faster. This thick segment, because it's so thick and it has no aquaporins, water can't move out. So water moves out on the way down, salts move out on the way up. You start with dilute solution up here. As water is removed, it becomes concentrated. So at the bottom, you have a concentrated filtrate. And then salts are removed on the way back up. And so many salts are removed that you end up with a dilute solution again, entering the distal convoluted tubule. So the distal convoluted tubule has about 20% of the water and about 7% of the salts remaining in it. If you peed out all of this, you'd be peeing out about 36 liters of water per day. Now that's a lot less than the 200 liters that you filtered in the filtrate, but if you peed out 36 liters of water per day, you'd be dead in a day or less. You don't have 36 liters of water to get rid of. So we're going to take that 36 liters down to like 1 or 2 in the distal convoluted tubules and the collecting ducts. Both the DCT and collecting duct reabsorb water. And the amount of water that they reabsorb varies depending upon what hormones are present. Aldosterone and ADH are going to promote salt and water reabsorption. So they will tend to keep water in your body, meaning that you're going to reabsorb more of this 36 liters. Natriuretic peptide is going to cause you to absorb less water, causing you to excrete more. There are 2 types of cells in the distal convoluted tubule and collecting duct. The principal cells, which are the most common, they have hormone receptors, and they're going to be the ones that are involved in whether or not you are reabsorbing salt and reabsorbing water. You also have intercalated cells. These are less common, and they are primarily responsible for acid-base balance. While acid and potentially base is excreted throughout the proximal convoluted tubule, primarily, most of the control over pH happens in the distal convoluted tubule. And it is a potassium acid, a potassium hydrogen antiporter. So you would reabsorb potassium and secrete hydrogen. In summary, the proximal convoluted tubule reabsorbs 65% of glomerular filtrate, including practically all of your nutrients, glucose, amino acids, things like that. A lot of water is reabsorbed here as well by osmosis. You're reabsorbing salt and other solutes. Where solutes go, water follows. So you are absorbing your solutes and salts and things like that through transcellular reabsorption, and then water follows through pericellular reabsorption. The nephron loop is going to reabsorb another 25% of that filtrate. Water on the downside, and then sodium, chloride, potassium on the upside. The distal convoluted tubule is going to reabsorb sodium and chloride and water, and this is where most of the hormonal control takes place. How much water you're reabsorbing is going to be controlled in the distal convoluted tubule and the collecting duct. How much calcium you're reabsorbing is going to be controlled in the distal convoluted tubule and the collecting duct. And remember that calcium reabsorption is under hormonal control. All throughout this system, you have active transport, secretion of drugs, waste products into the filtrate fluid, which eventually becomes urine. This is just a picture showing you what is absorbed where. So, as well as what the osmolarity of the solution is. You can see here in the filtrate, the initial filtrate has an osmolarity of 300. That's pretty dilute. By the time you get to the loop of Henle, it's still basically at about 300. As you go down the loop of Henle, the osmolarity of the filtrate increases dramatically. It becomes more concentrated, going from 300 to 400 to 600 to 900 to 1200 down here at the bottom. As you go down the loop of Henle, you are losing water, and urea is actually getting pumped in. As you rise through the ascending loop of Henle, your urine, your filtrate, I should say, gets less and less and less concentrated, not because water is being allowed back in, but because you are kicking out more salts, mostly with active transport. All right? So you're kicking out sodium, potassium, chloride through active transport, and by the time you get back up here to the distal convoluted tubule, your fluid filtrate is very dilute, even more dilute than the initial filtrate was, at around 200 to 100. Through the distal convoluted tubule, what you are reabsorbing depends largely on hormones. We'll get to that when we cover control. And then when you go down the collecting duct, you will be always reabsorbing urea. That's part of the countercurrent multiplication. We'll talk about that later on. And you may be reabsorbing water, depending upon your hormonal state. In the proximal convoluted tubules, you're going to be reabsorbing all of your various nutrients, glucose, amino acids, proteins, which shouldn't be there, vitamins, lactate, some urea and uric acid. It'll come in through it'll naturally follow the flow of water through the paracellular route. You're also going to be reabsorbing a fair number of salts, and you're going to be excreting urea, uric acid, creatinine, and some drug products back in. You don't want this urea and uric acid. That's the stuff you're trying to get rid of. But the cells here are leaky, so some will leak back in. In the descending loop of Henle, reabsorb water, secrete urea. Ascending loop of Henle, reabsorb salts. In the distal convoluted tubule, you can reabsorb sodium, chloride, H2O, bicarbonate. You can also reabsorb calcium, magnesium, things like that, depending upon your hormonal state. And you're going to be secreting potassium, acid, and ammonia. In the collecting duct, you're going to be reabsorbing water, maybe, depending upon your hormonal state, and urea. For this portion, I want you to know the glomerulus and glomerular filtration rate, what determines it, the four Starling forces, and how they work. You should know what gets absorbed where, all right? So if I say, should you find glucose in the descending loop of Henle? The answer would be no. All of the glucose should be absorbed in the proximal convoluted tubule. If I ask you, where is most of the filtrate reabsorbed? The answer would be the proximal convoluted tubule. 65% of it is reabsorbed there. If I was to ask you, where is most hormonal control exerted over reabsorption? That would be in the distal convoluted tubule. If I were to ask you, where is water reabsorbed through squamous cells due to an increasing salt gradient? That would be in the descending loop of Henle. So you should know, in general, what is reabsorbed where and why.